Good afternoon / evening, everybody. I'm EILA Skinner. I'm the chair of the department. I've got to meet a lot of you already. We are really excited to be hosting this. We've been thinking about it for probably 3 years while we're waiting for things to open up a little bit. So it's great to be here and to be outside with you. Welcome to the breakthroughs in prostate and bladder cancer, and we're going to focus on those areas. For background, I've been here for about 10 years, and I'm a urologic oncologist. I'm a surgeon specializing in bladder cancer. But we're not going to talk about me today. We're going to talk about my 4 fabulous faculty who are here with me. We're going to hear short presentations from each of the faculty members, just highlighting a small area of their research related to prostate and bladder cancer. After the presentations, we will have some time for questions and answers. And then there should be some time also to go back and visit the exhibits if you didn't have a chance to do that. We are going to be recording the presentations today, so you don't need to take frantic notes. We'll send each of you a link to the recording, and feel free to share it with friends or send us questions later if they come up. And we won't include the question and answer portion at the end, so if there's questions that are, want to necessarily publicize, you don't need to. And that you should be able to catch each of the faculty afterwards too if you have specific questions. So I'd like to first introduce our first speaker. Jeff SONN, who is here on my right, was my first recruit when I got to Stanford. Jeff did his urology training here and then went to UCLA for his fellowship and came back here in 2013. And he's just recently been promoted to associate professor, so we're very proud of him. His clinical practice has been focused on prostate and kidney cancer, and he's a phenomenal robotic surgeon, well known in that area. He also serves as the director of our Stanford MRI-guided prostate biopsy program, as well as focal therapy program. His research interest has been in that area, really trying to improve both diagnosis and treatment of prostate cancer, and focusing really on the interaction of imaging and patient care at the bedside. So that's kind of where his focus is. And he's the head of the Stanford Innovation Laboratory and principal investigator of a recent big clinical trial on prostate MRI-guided focal therapy of prostate cancer. So Dr. SONN, please come on up.
Thank you, Dr. Skinner. It's a pleasure to be here tonight speaking to you all. So let's dive right into some of my work. I think that many of you are familiar with PSA, which is a blood test that can be used for screening for prostate cancer in men to try to find cancers early before they have a chance to metastasize or spread elsewhere in the body. And this has been used for a number of decades. Now, early in the days, and for many years, a man who was found to have a high PSA blood test would go on to a biopsy procedure where a number of needles are placed into the prostate looking for prostate cancer. And I've referred to it here as a blind biopsy. And why do I say blind biopsy? Well, it's because it's usually driven by ultrasound. And this is a sample of a transrectal ultrasound image. And what you see is the prostate in the middle of the screen there. And this does a nice job of showing us where the prostate is, but a poor job at showing where the cancer is actually located itself. So we can't target a specific cancer, and instead what evolved is what we call a systematic prostate biopsy, which is essentially where you take 12 needle samples and evenly space them throughout the prostate. It's like setting up a search grid to look for prostate cancer. And as you might imagine, that can lead to a number of potential errors, such as that's depicted on the screen here, where you can see that blue spot meant to represent a prostate cancer that's missed just because it fell in between the biopsy cores of that grid. Now, on the other hand, if we do see prostate cancer, we find it on a biopsy, we often had fairly limited information about how big that tumor is, where it's located, and as a result, because we couldn't localize it well, we're really left with treatment options that address the entire prostate, either surgically removing the entire prostate or radiation treatment to the entire prostate. And those are good treatments. They remain the mainstays of treatment for prostate cancer even today. However, they very frequently can lead to long-lasting and life-altering sexual and urinary side effects of men who are undergoing treatment for prostate cancer, like high rates of permanent loss of sexual function and urine leakage. So the question is then, can we do better? And I would say the answer is yes, we can. And one way that we can really push this forward is more use of prostate MRI. This is a sample prostate MRI image that you see on the screen up here. The cancer here is this dark area off on the side, and as you could imagine, once you know that that area is where we're worried about, we can then guide biopsy needles into that area to reduce the chance of missing that cancer diagnosis. Unfortunately, though, not all prostate cancers show up this clearly. This is a prototypical case, but sometimes these things can be missed. And in fact, it takes quite a lot of skill from a radiologist to know how to find these things accurately. We published a paper a number of years ago that showed remarkable variation in the skill across a number of different radiologists in their ability to see and detect prostate cancer. So one of the big goals of my research group is to try to bring high-quality prostate MRI interpretation to more men throughout Stanford, throughout the country, even throughout the world. And there's a couple of different ways we could do that. One is we could train radiologists one by one to get better at reading these scans, which is important, but it's not very scalable. Another approach may be to try to train a computer to help a radiologist to do a better job at finding that. And that's a route that we've chosen to take through what we'll call machine learning, or essentially training a computer to help a radiologist find these cancers. And I'll say that AI, or artificial intelligence, can do this and has great potential to improve prostate cancer detection on MRI. But that takes a lot of work. Any AI challenge does. And what we need first is a lot of data. And in this case, what we need is a lot of prostate MRIs. And we've been working to build this up over the 9 years that I've been here at Stanford. We can see here on the left of the slide that 2 sample images from a man who undergoes a prostate MRI. So that's very important. We need these MRI images. But not only that, we also need labels. We need an answer key to tell the computer what does prostate cancer look like on these images so it can then find them on additional scans. And once we get all that information, the data and the labels, they can be plugged into a computer model, we'll call it a deep learning model, so that that model then can look at scans that it's never seen before and help identify prostate cancer. It'll spit out a heat map to tell us where in the prostate that it's worried about finding cancer that then can be provided to a radiologist reading these scans. Now, this takes a lot of work and a lot of skills, and so I've included pictures of a few people in the group that I collaborate with, although not from everybody. I want to highlight a couple of them. One, Dr. Richard Fan, who's an engineer that works closely with me and also within our department. He's here this evening. And also Dr. Mirabella Rusu, who's a PhD scientist, assistant professor in the Department of Radiology. And we all lead a group made up of postdoctoral scholars and research scientists that we hire to do this sort of important work that's only possible through grant support and philanthropic support to hire the people to do this important work. Now, in the last minute, I want to talk a little bit about treatment. So far, we just talked about diagnosis or finding these cancers. Well, what about treatment? Let's talk just briefly about breast cancer. Years ago, every woman with breast cancer would get a mastectomy, have the entire breast removed. But it was learned a number of decades ago that you don't necessarily need to do that. You can just remove the tumor itself and get great outcomes. And so too in prostate cancer, we're slowly moving to that paradigm shift of a cancer-centric approach as opposed to a whole organ-centric approach. And let me give you an example here. This is an MRI image of a man who I saw here, a patient of mine who I saw here at Stanford. And what we can see is this small black area pointed to by the yellow arrow, and that's his prostate cancer. And the rest of the prostate is totally normal. Now, he very much could have had a prostatectomy, had the whole prostate removed, or had radiation treatment to the entire prostate. But he wasn't interested in that. He wanted to try something that was less invasive and with a lower chance of side effects. So he ended up enrolling in a clinical trial using what we call MRI-guided focus ultrasound, which uses ultrasound to heat that area of the prostate where the cancer is located and kill it through heating. And this is an image immediately after that treatment. You see this black area here is an area where there's no more blood flow. So that area is dead, but the rest of the prostate is very normal. And so by doing this focal therapy approach, we're able to treat the cancer with the lower risk of side effects. This is very appealing to a lot of guys, but it does take years and many clinical trials to prove what is the appropriate role of focal therapy and to bring it to more men. So that's another big research focus of mine, is conducting these clinical trials with the end goal of bringing focal therapy to more men with prostate cancer. Thank you.
So save your questions. We'll do the questions at the end. And I want to go on and introduce our next speaker. Dr. Jim Brooks received his medical degree here at Stanford and then subsequent training at Johns Hopkins. Came back and joined the faculty in 1997. He's the Keith and Jan Hurlbut Professor of Urology. His clinical practice through his whole career has been focused on prostate cancer, and in recent years he's really been honing in on the whole area of active surveillance for early prostate cancer where we may not treat a patient right away, and Jim is one of the world's experts in that area. He's had an established basic science laboratory for his whole career as well. And in that, he really is looking at trying to discover the two, what we call a biomarker, some chemical or protein or something in the prostate that can give us a clue of what the cancer is how the cancer is going to behave and how it's going to respond to treatment. In addition to all that, he's had multiple leadership roles here at the school and is currently the vice chair of our department and chief of our oncology program. He recently also developed an interest in benign prostate enlargement, which actually is a very common condition and not very well understood. And he received a prestigious O'Brien Center grant to study that area, trying to apply some of the techniques that we've learned in prostate cancer towards that benign disease. So Dr. Brooks, I will let you tell us about your research.
My pleasure. Thank you, EILA. I felt a little sheepish when I came up with this title because in medical circles anyway, it's sort of become a cliche to talk about bedside to bench and back and things like that. But I picked it because it literally describes how we do research and how my lab has done research for now 30 years. Basically, we surgeons cut things out of people. So we take tumors out, deliver those to the laboratory where we investigate, try and make discoveries, many times using state-of-the-art technology not available elsewhere. And then from what we learn from that technology, bring it back, try and bring it back into the clinic. And that's the hard part. So what I want to do today is give you just two examples of this. There's many examples, but I want to give you two examples of projects that we've done where we've applied this paradigm to try and understand prostate cancer. The first is the use of genomics to understand these prostate tumors. This character over here is a guy I worked with, a biochemist named Pat Brown that I met not long after I came here, who had invented a technology called DNA microarrays. Now in the 1990s when I started doing research, if we were interested in genes in cancer, we'd look at one gene at a time and try to figure out what it did in the cancer. With Pat's technology, this is a printer that would make the microarrays, this is a microarray, we could look at all genes simultaneously. Now, when he invented that technology, he also had to invent a whole new way of doing statistics to try and understand the data and so on. So it really was paradigm shifting. It really blew the field open. So Pat is a disruptor, and he retired a little early from Stanford to go disrupt the field of food. He founded Impossible Foods and invented the Impossible Burger. So Pat's an incredible guy. So this is the type of data, this is a slice of the type of data that we would get from these microarrays. And think of this as like an Excel spreadsheet where each column represents a sample. The pink ones here are normal prostate, the colored ones are different cancer type, and each row here represents a different gene. And rather than having a bunch of numbers in the Excel spreadsheet, we paint the cells with color. So red means a gene is cranked up. Green means it's turned down. And we use a variety of statistical approaches to pull out genes that we think are correlated with more aggressive cancer or less aggressive cancer. Then we test them on tissue microarrays. And we have a little display what tissue microarrays are back there, where we can look across a whole bunch of samples and find out if that gene connotes good or bad cancer. So we've been doing this iteratively for many years now and identified a set of bad genes that connote aggressive prostate cancer. And we've published these and put them out in the world for use. Now, scientists get excited with lists of genes like this or identifying genes like this because they want to understand how these genes work to drive cancers and make them behave aggressively. I'm a clinician. So for me, I have clinical questions. Let me give you an example. With PSA testing, we diagnose 300,000 cases of prostate cancer every year in this country. 30,000 men die of it. So there's a big discrepancy between the number we diagnose and the number destined to die of prostate cancer. Why is that? Well, the reason for that, we've known since the 1940s that there are a lot of guys walking around with prostate cancer. If you do autopsies on men who die of other causes, heart attack, stroke, car accident, and have a card-carrying pathologist slice that prostate up thinly and look in it, these are the numbers here. For men in their 60s, about 2/3 of them have cells in their prostate that are prostate cancer cells. 2/3. 3% of men die of prostate cancer. So what that means is there's a lot of prostate cancer out there to be found if you start doing PSA testing and biopsying people. So what do we do with that? Well, I just sat in clinic yesterday and talked to guys who had what looked like low-risk prostate cancer, but I don't know. I'm trying to figure out which one's the tiger and which one's the pussycat, right? So that's what these genes can be useful for. And some of the genes that we have found now are in clinical tests. So these are 3 different clinical tests that are out there that can be done on a biopsy to genetically profile them and tell you whether or not the cancer is aggressive. And there are a bunch of genes we discovered in those tests. We didn't patent any of them. Like I said, we just dumped it out there. And it's thrilling to see that they're used. The other thing we've done is now that these tests, we're developing these tests and have these tests, we've put together a large group of patients that look for all the world like they have autopsy cancers, but we're not sure. About 40% of them will become more aggressive in 5 years. And so now we have about 2,100 patients enrolled across the country that we collect blood, urine, biopsy samples. We now have hundreds of thousands of samples that we can use to test these markers to see if they actually work to predict. So this is project 1. Second project I want to talk about is one that's just a few years old, and I'm really excited about it. This is a cell. This is over on the side here. This is an electron microscope view of a cell. This is the cell membrane down here, and this kind of lawn on top of it, this grass on top of it, is actually strings of sugar. Cells are literally sugar-coated. And the sugars are attached to proteins, hence the name glycoproteins. And it turns out that these sugars vary between non-cancerous and cancerous tissue. They differ. And recently, there's new technologies to look at that. I work with a chemist, Carolyn Bertozzi, and a protein biochemist, Sharon Pateri, to do that. This is an example from prostate cancer. So prostatic acid phosphatase was the old-fashioned PSA. And you can measure it in the bloodstream. On the bottom here, one of these spots on prostatic acid phosphatase, these are how a chemist draws sugars, these long chains here. There's only 2 of them. In cancer, there are 7 different types of sugars at this spot. Imagine if a patient comes in, has an elevated PSA, and now I measure this and say, whoops, there's all these new sugars on here that look like cancer. This is a patient I need to send to Jeff for imaging. This is a patient that might need a biopsy, right? Whereas the other guy could say, elevated PSA, but it all looks like normal sugars on it. We can ignore that. So we need enabling technology to do that, but that's where we're going. Another thing we found, which is really interesting, is there's a different type of sweetness in cancer. These little diamonds on the end connote what's called sialic acid. It's a type of sugar that really goes up in cancer for reasons I don't quite understand. The reason sialic acid is interesting is this. Sialic acid, this little figure here, shows it binds to a receptor like a key in a lock on immune cells and turns them off. So this sugar can turn off immune cells. It's remarkable. This is analogous to the work Jim Allison did that won the Nobel Prize a couple years ago. This is immunotherapy. So what Jim Allison discovered is that there's cancer cells, devious things they are, make PD-L1, which is a protein that also turns off immune cells. If you interfere with that, suddenly the immune system can recognize cancer again. Glycoprotein, sialic acid, is doing the exact same thing. So we're running very fast. We have data in our laboratory that this is operative in prostate cancer, and we're running trying to see if we can get it to the point where it's translated into the clinic as a new therapy. So I gave you examples of how we take tumors and analyze them in the laboratory, then try and bring it back. So bedside to bench, bench to bedside. Happy to discuss with you any other example. We have several other examples where we've done this in other tumor types and in prostate cancer. And I want to thank you.
Thank you, Dr. Brooks. That was fantastic. So our next speaker is Dr. Joseph Liao. Dr. Liao also received his medical degree here at Stanford and then did his urologic training at UCLA and joined the faculty in 2006. He's now professor of urology, and he's currently the vice chair of academic affairs and director of research in our department. Dr. Liao maintains an active clinical practice at the VA, Palo Alto VA Medical Center, and he has been the chief of the urologic division of that organization for the last 15 years, just recently passed that on to another faculty member. In that role, he was responsible not only for the urologic care at the Palo Alto VA, but also at a bunch of outlying clinics, so it was a big job. He has, throughout this, maintained a very active laboratory as well, but focused a little more on bioengineering, kind of between Dr. Brooks and Dr. SONN, I would say. And he is really, part of his focus has been in bladder cancer. So we're going to shift focus now and talk for a couple of moments about bladder cancer. And he has been using both advanced imaging, using some of the engineering skills that he has, and you probably saw some examples of that, and also looking biologically at the urine. What sort of things can we discover in the urine that can actually tell us what's going on in somebody's bladder cancer? So he's going to tell us a little bit about all that. Thank you, Dr. Liao.
Thank you, Dr. Skinner, for that very kind introduction, and good evening. Over the next few minutes or so, I'd like to share with you the research in my lab on how we can try to improve bladder cancer treatment through augmented imaging and liquid biopsy. A little bit of background on the current paradigm for bladder cancer diagnosis and treatment. Patients typically present with blood in the urine or hematuria. That usually then leads to a consultation with a urologist. Where in the office setting, we put an instrument called a cystoscope inside the bladder so we can inspect the lining of the bladder to see what the potential causes of the bleeding is. If a tumor is found, then the patient goes to the operating room where we do a procedure called transurethral resection of bladder tumor, or TURBT, to remove the tumor. If the patient is found to have non-muscle invasive bladder cancer, and commonly a course of treatment called BCG, which is a topical treatment that we put inside of the bladder, is a form of a localized immunotherapy, and that's going to be followed by surveillance cystoscopy up to every 3 months, because bladder cancer has a very high recurrence rate. If, however, patient is found to have muscle invasive bladder cancer, then the first line treatment is actually a course of chemotherapy, followed by removal of the bladder, a procedure called radical cystectomy, and the reconstruction of the urinary tract. As you can see, the treatment algorithm is quite complex, and at really every step of the way, accurate diagnostic information and staging is critical, because all of these treatments carry a share of side effects and potential complications. So the focus of my lab is to address some of the major unmet needs in bladder cancer. We want to develop ability to see the cancer better. We want to develop ways that we can actually detect the cancer at the earliest stage non-invasively. What about when surgery, BCG, and chemotherapy fail? We want to actually develop new treatment. And not only just to develop new treatment, we need to actually develop ways that we can follow the treatment. Is it working or is it not working? Do we need to switch? Those are the things that's driving the research in my lab. On the right-hand side of your screen, you can see that bladder cancer comes in all shapes and sizes, and sometimes it's actually difficult to tell where the cancer is within the bladder. In fact, cystoscopy can miss clinically significant bladder cancer in up to 20–30% of the time. And we know that TURBT may leave residual cancer behind. There are new technologies such as blue light cystoscopy which can help us see better. However, it's not widely available and is somewhat cumbersome to use. We believe the answer lies in artificial intelligence, and we're using state-of-the-art tools through computer vision and machine learning to help us see the cancer better. We developed an algorithm called CystoNet for that purpose, and what we did in this study is that we took 100 patients with biopsy-proven bladder cancer, a type called papillary urothelial carcinoma, which is the most common type of bladder cancer. We put it in a training set and a test set, then apply it to another cohort, patient 50 or so, with and without bladder cancer. And for the first time, we were able to actually track these cancers dynamically and with really great performance status. And we're just very much getting started. Now we're actually putting this in the operating room where we see how it can help the urologist in real time. What type of user interface do we need? We're developing a second generation of CystoNet in which now we're amassing a large quantity of bladder images and videos from the full spectrum of bladder cancer, as well as other cancer-mimicking benign diseases that are found in the bladder. And what we hope to do is to develop a predictive model that integrates your medical record, your clinical history, imaging, and then biomarker information. And we believe urine is the answer there. Urine has long been recognized as a window into your health for thousands of years. It is non-invasive, it is abundant, it's easily accessible, and is very amenable for longitudinal monitoring, and we believe it is really the ideal source in this era of precision medicine to follow bladder cancer. Using these new strategies, what we're trying to do is we have now developed 2 different but complementary urine tests to detect the messenger RNA from the pellet of the urine and the cell-free DNA or tumor DNA from the supernatant part of the urine. In this first test, what we did is develop a simple rapid urine test to detect a panel of 3 different biomarkers in urine using a 2-hour PCR test. So this is a lot in the news recently. It's really very similar to the COVID test we've been all taking recently, and it requires a couple of teaspoons of urine. What really makes me exciting about this research is that we can, for the first time, do what I call molecular triaging. That is, with a small amount of urine, we can actually tell you, are you at high risk for bladder cancer or a low risk for bladder cancer? Do you need to do a cystoscopy? Or can it be delayed or avoided altogether as opposed to somebody who needs to expedite cystoscopy and TURBT? And in this recent study, we found that you can actually obviate the need for cystoscopy in over 50% of the patients. In this next test, what we're developing is a ultra-sensitive way to detect urine tumor DNA that's found in urine. And this is done in collaboration with a colleague at Stanford, Max Dean. And what we're doing here is to do what is called targeted sequencing of parts of the DNA that's mutated in patients with bladder cancer that's shed into the urine. What's really exciting for me about this assay is that it's ideally suited for treatment monitoring. So on your right, you can see a couple of representative patients. On the top, this is a patient who's undergoing BCG treatment, that localized immunotherapy that I talked about. And the amount of urine tumor DNA perfectly correlated this patient's clinical status is that he has good response to BCG, has no recurrence. In contrast to the patient on the bottom, where clearly BCG is not working, recurrence is happening, and it's time to switch to a different type of therapy. And we have ongoing studies to apply this technology, not just in patients undergoing BCG, but also chemotherapy, as well as some of the newer systemic immunotherapy treatment options out there for bladder cancer. So this is sort of the grand vision. Our mission is to develop precision diagnostics to guide precision therapy of bladder cancer. We believe in early detection, risk assessment, and monitoring your treatment response through integration of state-of-the-art biomarkers, imaging, bioinformatics, and AI. And perhaps in the not-so-distant future, a patient at Stanford and elsewhere can get something like a personalized cancer dashboard in which we can plot out the different time points, overlay that with the biomarker assays that we're developing, and use that to guide the timing and the need for cystoscopy. And together with all the other data, your clinical history, your other risk factors, et cetera, to really develop a personalized predictive model for you, the patient. Lastly, I just want to thank the patients who participate in the research that we do, because without the patients, this is not possible. I also want to thank the folks in my lab who carry out the bulk of the work, and I'm humbled to be their leader of a passionate group of clinicians, scientists, and engineers. And thank you for your attention. I'll be happy to take any questions later. Thank you.
I expect you're seeing some themes here that kind of tie all these different research projects together. I think that here at Stanford, the incorporation of artificial intelligence and machine learning is reaching, I think, every part of healthcare. And you know, we can all see, I think, the point in the near future where that will become really critical to how we treat patients. So my final speaker for tonight is Dr. Philip Beachy. Dr. Beachy received his PhD from Stanford, and after a time at Johns Hopkins, he joined the Stanford faculty in 2006. He ultimately joined the urology faculty just a few years ago in 2018. He holds the Ernest and Amelia Gallo professorship in urology, and he was a Howard Hughes investigator for 30 years, long time. Dr. Beachy is director of the Siebel Investigator Program at the Institute of Stem Cell Biology and Regenerative Medicine. He is, and Dr. Beachy, up till now, you've mostly been hearing from clinician scientists. Dr. Beachy is like a real scientist. He is a brilliant scientist. So Dr. Beachy is best known for being one of the first people to describe both the function and the biology of something called the hedgehog pathway, which I hope you'll tell us a little bit about. It's one of the real keys to both normal development and actually a key to a lot of cancers and how they behave. He will tell you how this impacts bladder cancer and some of the exciting things that he's doing now in the lab. We are thrilled that he has turned his sights on bladder cancer. I think this holds a lot of excitement for the future. So I'm going to let Dr. Beachy take us from there.
Well, thank you, EILA, for that introduction. I really consider my clinical colleagues the real scientists. They're the people who are having direct impact on people's health. It really is a pleasure for me to be here and have the privilege of speaking with you all. I'd like to discuss two of the major challenges in bladder cancer. And these are the challenges of recurrence and progression. Now, as you may know or may have gathered, the great majority of bladder cancers at diagnosis are non-invasive. They're restricted to the lining of the bladder. They don't enter the tissues, the cells that sit underneath. And so they're non-invasive. Nevertheless, with this kind of superficial tumor, when it's removed, even if it's successfully removed surgically, there is a high probability of recurrence, a very high probability, and this is because the remaining, tumor proximal or even cells distant from the site of the tumor have they're more susceptible to the occurrence of a new tumor, and this is recurrence, and this is the problem of recurrence. Another issue in bladder cancer is that non-invasive cancers, most of them at diagnosis, can progress to invasive cancer. And if you have an invasive bladder cancer, this usually means a major surgery, which carried out by one of my colleagues, which can be life-saving, but it's also life-changing. So how can we stop recurrence and progression? My lab's approach to this is to learn more about the basic biology of bladder cancer, and this actually begins with learning more or understanding as much as possible the basic biology of the bladder. And so beginning here with the lining of the bladder, what you have at the innermost lining, the innermost surface, are these tightly linked very large cells called umbrella cells, and they're called umbrella cells because they shield the cells sitting underneath from the liquid wastes that accumulate in the bladder. Then you have a layer of intermediate cells and a layer of cells at the basal portion of the lining. We actually call them basal cells. They include the stem cells. And these cells are very important in providing renewal for the tissue. This is required because of day-to-day wear in the bladder or because of injuries that may occur. For example, urinary tract infections, which occur in about 10% of women annually. So these basal cells or stem cells are required to produce the other cell types in the bladder. And they're a bit really like a person with a liberal arts degree who is not yet specialized for a specific job, but could become eventually an attorney, a politician, a civil servant, an artist, an entrepreneur, even a doctor or an engineer. So a stem cell is an unspecialized cell that can make cells that are specialized, but a stem cell, to maintain this renewal capability, also has to make more of itself. So in the context of cancer, this sounds a little dangerous, right? A cell that makes more of itself. And indeed, it is dangerous. We know, and I'll say more about this in a minute, that stem cells are actually the source of bladder cancers and of a number of other cancers as well. So how can we study this? Well, we need some really good tools to look at these things, and we have them in the mouse. And one tool that we have to figure out what cells will do is to mark cells genetically and then to trace their fates. And so this is an example of that. This is the rainbow mouse. You can label cells with any of these 4 colors. This shows an example of a bladder muscle where cells are randomly labeled with one of these 4 colors, but then that cell is permanently labeled and all of its progeny, all its descendants, will retain that color. So you can track that cell. In the mouse, we also have a way to induce cancer. We give them in their drinking water a carcinogen like the carcinogens in cigarette smoke, which are the major risk factor for bladder cancer, okay? And after a certain period of exposure to this carcinogen, we see this kind of thing in these mice. We see that a single cell, a yellow cell here, has taken over the entire lining of the bladder. It has grown and spread. This is another example, another mouse, another bladder, a different colored cell, blue here. But in both these cases, neither the yellow nor the blue cell has invaded to the tissues that lie underneath. After a bit longer exposure, in a different mouse, different bladder, different color here, you can see that these red cells, which are the cancer, have become invasive. They've taken over most of the bladder. And the nice thing about this model is that it very cleanly separates this early non-invasive stage from the later invasive stage, and that allows us to study this process. Okay, so that helps us with studying progression to invasion. Now, I said earlier that cancers come from basal stem cells. How do we know that? Well, again, these are some of these powerful genetic tools. Genetic marking, again, we mark the basal cells in the lining of the bladder, which normally would make intermediate cells and umbrella cells. However, in this carcinogenesis model, what we see is that the tumors that arise are all the same color as that label, which means that they're arising from those labeled cells. And that's how we know that these basal stem cells are giving rise to cancers, not only in the bladder, but in the colon, pancreas, and a number of other cancer types. So these are mouse studies. How do we learn about progression and recurrence in human bladder cancer? Well, I'm fortunate to have these wonderful colleagues and others in the department here who provide us with very precious patient samples. And these are essential for understanding how the human biology works, the biology of human cancers work. And so to study progression, what we do is we compare non-invasive cancers to invasive cancers, okay? Makes sense. Try to see what the differences are. To study recurrence, what we want to do is to compare a completely normal bladder tissue to a tissue that comes from a tumor-adjacent part of the bladder, or even tumor-distant. This is a quote-unquote normal, or we call it sometimes apparent normal, portion of the bladder. And by comparing these two, we can maybe understand why it is that apparent normal tissue is so much more likely to produce a tumor than a normal tissue. How do we carry out these comparisons? Well, we've already heard about genomic approaches that Pat Brown pioneered here at Stanford. Things have actually evolved even further. We can look at, as Jim mentioned, the entire genome, thousands and thousands of genes all at once, but we can look at them in individual cells, one cell at a time. And so we do this by a technique called single-cell RNA sequencing. And this is how we carry out these comparisons, and I just want to quickly show you an example of this. This is a representation of 9,000 tumor and normal cells in one experiment. Each dot here, you see many dots. The software that works with these data is actually called Seurat, the impressionist who used pointillism to represent beautiful scenes, at the beach or at a picnic. Each dot is a cell, and each cell is associated with about 50,000 different RNA sequences. And what we can do is to use those sequences then to compare these cells to each other. And greater similarity is indicated by proximity here. So cells of the same type are clustered. And this big cluster up, actually several different clusters, are all immune cells. Down here are cells of the blood vessel wall. I've circled here cells that come from the lining of the normal bladder. And then here are some cells from a tumor, which also arises from the lining of the bladder. You can see that they're clustered far apart because they're different. And in fact, we see differences in the clustering behavior of a tumor that's invasive and a tumor that's not invasive. Okay, so this is single-cell RNA sequencing, and we've applied this in order to study recurrence. We've applied this to normal bladder tissues and apparent normal bladder tissues that don't include tumor but come from a bladder where there has been a tumor. And so these red cells are the normal, the green cells are the apparent normal, and these are subclusters within those cells. And now if we look at, if we look at the expression of a gene, a specialization gene, which is indicated by the purple color here, you see that it's most prominently expressed in the normal cells and not much at all really in the apparent normal cells. On the other hand, if you look at a gene that indicates stem cell or basal character, you see that it's expressed at highest levels, the brightest purple here, in the apparent normal cells. And it's there, but not nearly at as high a level or in as many cells in the truly normal tissue. So could it be, is it possible that the higher risk of occurrence of a new tumor is because these apparent normal cells have greater basal or stem cell-like character? After all, I told you that it's stem, and I showed you that it's stem cells that are the source, the origin of these tumors. And if that's true, if that were true, would it be possible to block or slow recurrence by reversing this basal character that these cells can acquire? Okay, so I've been showing you these studies in the mouse, in humans, I mean. Now we need to go back to the mouse to try to answer these questions. Can we reverse basal character acquisition, and does it help? So what we're doing here in the mouse is looking at two basal genes, never mind their names, one's in green, one's in red, and this is just a normal mouse. If we give them another mouse the carcinogen that I talked about earlier, you can see that expression of both of these basal markers increases dramatically. But if we treat those mice with drugs, two drugs actually, that we chose because from our work and the work of others, we thought that they might be able to reverse this, the acquisition of this basal character. And indeed, that turns out to be true. So there's less green here in this with drug 1, less green with drug 2, less red, and so forth. And in this graph, you can see here, this is more than 2 genes now. This is just a whole set of genes that are considered together to create a basal score. This carcinogen-treated mice have a very high basal score, but that is reverted by treatment with these 2 drugs. So that looks interesting. What's happening with the bladders? Well, here you see normal bladders, control bladders, and here you see bladders from a mouse that's treated with the carcinogen but no drugs. And these bladders are much bigger. You can see here that they weigh more. and the red dots indicate that they are full of tumors. These bladders here are from mice that were treated with the drugs, and those are these bars over here. They look much more like normal, and there are no tumors. So this is very interesting because drugs 1 and 2 act in the same biochemical pathway. So these results are mutually reinforcing, and because both of these drugs are FDA approved for treatments of other cancers, they could be used conceivably in trials with patients to see if we can block the recurrence of bladder cancers once you remove the initial cancer that's seen at diagnosis. So we're very interested in carrying out this kind of trial. We're also interested more broadly in obtaining this kind of information for every patient that comes in. If we can collect these kinds of single-cell data from apparent normal tissue and from tumor tissue, and then, of course, we have to treat the patients, and so we would deliver appropriate therapy based on clinical considerations. This would include surgical excision, perhaps BCG, which Joe has mentioned. Maybe even if our trial works, specialization therapy. Another thing that we get out of this kind of single-cell sequencing is that we may be able to select the appropriate immunotherapy. Immunotherapy has been very effective in a number of cancers, and it's just now beginning to use increasingly in bladder cancer, but it's not always clear whether it will work or which kind of immunotherapy should be used. We should be able to judge that based on the single-cell RNA sequence. And so we'd like to collect all this data, correlate the treatments with outcomes for individual patients, and after we gather a certain amount of information, we're going to be able to devise an individualized treatment plan for each patient when they come in at diagnosis based on this kind of very, very rich data that we have and that we can accumulate. So I'll end here by acknowledging Chris Prado, who's the newest member of our department. He was just offered a job as an assistant professor, which he has accepted, and he did the experiments in my lab with drug 1 and drug 2 that I discussed. Hua and Shuo are here this evening, and they are postdoctoral fellows in my lab working on various aspects. I've only talked about a couple of the projects in the lab, but they're part of the bladder team here. And Mallory is a graduate student who's also working on bladder as well. So with that, I'd like to thank you for your attention, and I'll be happy to take questions whenever.
That's fantastic. Department of Urology, I think, is poised to really pave the way for leading a number of breakthroughs in these diseases and to incorporate these discoveries, especially into clinical care. Our ability to accomplish this is, to some extent, really dependent on our donors, and we do want to recognize those of you who have helped support the department and support the researchers in the department, and thank you very much. And if any of you are interested in learning more about how you can support us, Evelyn, are you hiding in the back? She's our go-to person in the white jacket, so please feel free to grab her. So thank you so much, and appreciate your attention.